Chattanooga, Tennessee, Professor Joey Shaw, and I'm going to let him tell you a little bit about himself. And we've been spending money with the Ozark Chinquapin a lot in the field work. And last night our board, we were talking about and brainstorming about some of these other things we need to have done. And AJ Hendershot was talking about some of the money we were putting in to the analysis right now as we speak in California. If they haven't cashed it, they're about to cash a check for $1,395 for never before done work on the food analysis of the Ozark Chinquapin. And so that's a real important part. The other part, standing right beside me, is Joey Shaw, and on not even a shoestring budget, maybe a dental floss. He's been working, and out of his own gratitude, he has done stuff for us we would have literally had to pay thousands and thousands, I know for a fact, to have done, and he did it on his own. And he's really excited about his findings, and we're going to be getting him, I know, $1,200, maybe a little bit more, depending on how it goes. We hit a little technological glitch that we're going to work around. And so anyhow, we're really glad to have Joey here. We've been trying to get you over here for about 3 or 4 years, and hopefully if we treat him good enough, he'll stay here. And Joey's one of these people that is doing what he is doing, not because of money, but because it's unknown information. And last night we told him, I said, you're like on the head of the spear, just like Colton Zirkle back there. You know, you're taking what was thought before to be the knowledge about how something's supposed to be, then you get the facts and then it actually changes it. And you know, if you got good information and you know, you check all your facts, you're real solid on it, then you can change everybody's thinking on it. And I remember when I first started working with the Ozark Chinquapin, you know, it was hard to get very many people very interested in it. And now it's like a gimmick. You know, we see the benefit of food, wildlife, and diversity of our forest, and everybody's on board with it though. And so, you know, my hat's off to everybody like you and Colton back there, and I'm going to give it to you. Here you go.
Thank you, Steve. Grab this. So, my name is Joey Shaw, and like Steve said, I'm from Chattanooga, and I brought a couple of students with me. In the back is Taylor Perkins, who's a graduate student in our department. Yeah, he's back here waving.
They need to be up here.
They're okay back there. Yeah, we spent 12 hours in a car. They're good back there. So Taylor did an undergraduate honors thesis working on American chestnut, and Jesse is an undergraduate now who's coming into this project. And so both of these guys have been helping me with work over the last year or two. I also put on this talk this afternoon, I call her my academic sister, Ashley Morris, and she's at Middle Tennessee State University now. It's good to have her right down the road. She and I started graduate school together and have known each other for years. And I'll tell you a little bit about some of the stuff that we're doing and how it's relevant to work on Ozark Chinquapins. So I'm not going to go through all of this research. I've given a talk to you guys for the last couple of years where I really talked you through a lot of the genetic findings of the American chestnut, the Ozark Chinquapin, the Allegheny Chinquapin, but I'll just sort of rehash a little bit about it. I got involved with this project because when I started my position in Chattanooga, there's a guy there by the name of Hill Craddock, and he is deeply invested in the American Chestnut Foundation. And so he came to me with a problem, and he said, you know, we've got these what he thought may have been hybrid chestnut Chinquapins just south of Chattanooga, and he wanted me to look into those with the genetic tools that I use. And so what we did was this study that we published in 2012, but it was really focused on what's going on in the southern Appalachians around Chattanooga, and what are these weird morphological trees that appear to be hybrids. So what we did was we focused our sampling on chestnuts and Allegheny Chinquapins, and we sort of threw in, as you see here, we threw in a few token Ozark samples. And so what we ended up doing was we sampled American chestnut throughout its range, and we sampled Allegheny Chinquapin throughout really its range. We sort of had a gap here. We sort of poor sampling, the way the study kind of fell out. But we had Allegheny Chinquapin sampled and Chestnut sampled, and like I said, a few sort of token Ozarks. And what we did is we generated this phylogeny. Now, phylogeny is a hypothesis, right? It's an idea of the evolutionary relationships of the species. And so what I generated for that study was this phylogeny. And this shows, and you've seen this figure before, I've seen it on several of the posters around the room. And so really what this figure is showing is that all of the American chestnuts fell out here in this clade in two different genetic types. We call them haplotypes. All of the Allegheny Chinquapins that were really from the Appalachian plateaus, right, the Cumberland Plateau area, and really sort of the southern Appalachians here, they've got a distinct morphology. The leaves are kind of small, they're really white, tomentose, or hairy underneath. And that morphology fell out right here as sister to the American chestnut. And then what was really sort of unexpected was we ended up with all of these genetic types in this clade, which turned out to be all of these Allegheny Chinquapins, but down here from what the coastal plain, mostly the coastal plain, but kind of bleeding up into the southern Appalachians a little bit. And it's really sort of interesting to think that there's, you know, and then here your Ozarks, right? And so the Ozark Chinquapins have these completely different haplotypes. And so it was really sort of an interesting and unexpected finding to find 1, 2, 3, if we trace all these back to here, 3, 4 genetic types, 4 independent genetic lineages for what we've been recognizing as 3 species, right? American chestnut, Allegheny Chinquapin, and Ozark Chinquapin. And so that was the gist of all of this work, and that's how you guys sucked me in, or how Steve sucked me in to the Ozark group here. As he called me up, and actually while we were doing this, we contacted him and we asked him for some samples from your area, and he was happy to send them. And so over the last several years, he's been sending, and all of you have been sending many more samples my way. And so that's really what I'm going to talk about for the next few minutes, is just how can we design this study over the next year or two to be as solid as possible, okay? So, what are the questions that I think we share? What are sort of my research questions that'll satisfy my academic curiosity, and what are questions that you have about the Ozark Chinquapin, and you know, what questions do we sort of have in common that we can work together on? And so I'm thinking about the questions for the next phase of study. Well, question 1, what are the relationships between North American Castanea species? Well, what I showed you here, this phylogeny does show some relationships, but it's incomplete. It's incomplete in the sense that this is all from, get this fly buzzing around my head, I'm sorry, it's distracting me a little bit here. This phylogeny is all from maternally inherited genes, okay? So it's basically like if you wanted to know where did you come from, right, and somebody did this genetic testing on you, but only looked at your mother's half. And so what we need to do is we still need to sort of repeat this kind of study, but using different genes, genes that come from the nucleus rather than genes that come from the chloroplast. And that'll give us sort of both halves of the sort of family history of these species. So what are the relationships between North American Castanea species? Well, we're still working on that. How did North American Castanea species come to inhabit their present distributions? How genetically diverse is Ozark Chinquapin? How is the genetic variability of Ozark Chinquapin distributed throughout its range? And I think that question is very applicable to the work that you're doing. And can the data generated from investing these questions up above guide your conservation efforts of the Ozark Chinquapin? So, getting at question number 1, what are the relationships between North American Castanea species? Well, to get at this question, we use sort of a tool, a discipline that we refer to as phylogenetics. And that is basically building phylogenies, or building evolutionary histories for groups, right? And so to do this, what we do is we use genetic clues, or shared characteristics, to build hypotheses or models of the evolutionary history, or to build these phylogenies. And so we talked about that. And that discipline is called phylogenetics. How did North American Castanea species come to inhabit their present distributions? Well, to get at a question like that, we use tools from the discipline that we refer to as biogeography. And biogeography is the study of the distribution of species across geographic space and geological time. And so what we do is, again, we can use genetic clues or fossil evidence. We can use common migratory pathways, climate modeling of the past and present, to build hypotheses or models of past, present, and future ranges. And we've talked about some of that stuff today when earlier this morning we talked about the distribution of Castanea pollen throughout, from the last glacial maximum to the present. All right, so we use tools from biogeography. How genetically diverse is the Ozark Chinquapin? Well, to get at a question like that, we'd use tools from population genetics, which is the study of the distribution and changes in gene frequencies in populations. And so what I'm thinking about for our work together is to use more dense sampling within populations to determine frequencies of genetic types within the population. So if you look at, if this larger circle then represents the whole range of Ozark Chinquapin, and we look at several populations, and within several populations you can see there's a difference in the genes that make up this population, or not a difference in the genes per se, but a difference in their frequencies that make up this population versus this population versus this population. Now, what if it turns out that we learn through our collaborative work together that you can identify more blight-resistant strains sort of are tied to this orange genetic type, then that can sort of guide your conservation efforts in the future. And so we'll pull in tools from population genetics. And lastly, phylogeography. The discipline of phylogeography is something that I'm particularly interested in. And it really pulls in tools from all three of these other disciplines. So phylogeography is basically building a phylogeny, building an evolutionary history, and mapping it to the distribution of the genetic types over geographic space. Right? So this is combining population genetics level sampling with building evolutionary models and comparing that to species presence through space and time to really reveal historical spread and the distribution of genetic variability throughout these populations over the landscape and throughout time. Right? So it's really sort of big picture stuff. Here's just sort of an example, a really simple example. When you look at this fish, and you look at the phylogeny, and here's this phylogeny, this evolutionary history of this fish, and these different genetic types. And when you apply that in a phylogeographic context, you map that to the geography, you can see that this clade then, with the genetic type represented by a square, has a distribution here, right? And this clade then, with the genetic type, these black circles, has a distribution here, and so on. And so it allows you to start thinking about evolutionary processes and the geographic spread of different genetic types. And this is the sort of thing that I would like to get to with the Ozark Chinquapin. Okay? So I want to give you sort of an example. Here's a paper that was, or a figure from a paper published by my friend and collaborator Ashley Morris. This is on sweetgum. And what she did was she sampled about 5 specimens per population. Now each of these dots represents a population, and you can see that the populations are distributed throughout the range of sweetgum. And each different color represents a major genetic type. Right? So when you look at this, you can see that this genetic type that she's sort of indicating by a blue color is really represented here in the east. and you don't find that genetic type out here. And you find some reddish ones up around here and down in the south. And this is the kind of sampling that I would like for us to get for Ozark Chinquapin. And the reason for that, and not just collecting these plants, but I also want you guys who are the boots on the ground out here in the Ozarks, not to send just leaf samples to me, but also if you've made notes on each of these samples with respect to your interpretation of blight resistance, for example. Or what are the morphological characteristics that you thought about when you saw this thing in the field? Are the leaves particularly, are they hairy or they're not hairy? Or are they those large 10-inch leaves that we heard about this morning? Or are these smaller leaves or whatever? And I would also like for you to collect ecological data. We've talked all day about different soil substrates and elevations and physiographies. And so if we have notes about all of those different things that we could tie to these populations, it may be that we can run some numbers and figure out something like, oh, well, it's this orange type that's particularly blight resistant, and it's sort of spread throughout the range here. And again, that may be able to guide your conservation breeding efforts in the future. So the truth is, there really aren't that many studies like this in the eastern United States, and adding Ozark Chinquapin to the small list of these kinds of studies will really bring attention to the species from a lot of researchers and conservationists. So what I'm going to tell you about for just a minute is a review paper that this isn't published yet, but this is a review paper that Ashley and Charlotte Germain Aubrey, who's a postdoc down at the University of Florida, and I have been working on for, about 6 months now. And what we've been doing is we've been reading as many papers as we could get our hands on with respect to phylogeography. And this chart right here, this graph is showing the number of studies, so 1,200 studies overall from 1987 to 2013. We've got a couple of years left of data and papers to sift through to add to this. But what I really want to show you is that the number of papers over the last couple of years is really great compared to what was done just 5 or 6 years ago. Also, this tan color represents the number of non-plant papers. So frogs or fish or fungi or mammals or whatever. And so plant phylogeographic papers really only make up about 11% of the total. So this is really sort of new territory when you think about plant phylogeography in eastern North America, and putting together a sampling strategy like I'm talking about and putting together a study like this for Ozark Chinquapin will really sort of elevate the stature of this, like I said, among researchers and conservationists. One of the other things we were looking at in this review paper is what genetic markers do people use and why. And I won't get into the why, but I'll show you that roughly 50% of all of the studies that we looked at were using chloroplast sequences, organelle sequences, the same kind of sequences that I've used for this and the same kind of sequences I've been generating out of the samples that you guys have been sending me. So, we're right in the mix with that sort of thing too. Once data like these are collected, and if we have information on the genetic type from within the individual and where that individual was collected, when it was collected, and the, some ecological notes and all these kinds of things, there are other studies that have come out that show some really cool things. You could start looking throughout the population and start thinking about barrier analysis, right? What are the barriers to gene flow within your species? You could generate heat maps showing the spatial analysis of the genetic types. And so there are lots of really cool things you can do, but you need a big sort of robust data set with lots of information, not just information on what genetic type and where it was collected. Okay, so that's sort of what I wanted to address today and sort of push us in our collaboration.